For most of modern history, humanity has told itself a simple story about its beginnings. A single origin, a slow expansion, one ancestral population giving rise step by step to everyone alive today. Differences were temporary. The separation was shallow in the deep past, we believed. Early humans were never truly divided, but the past does not remember itself the way we do. Beneath the soil of caves and riverbanks, beneath layers of ash and collapsed stone, fragments of human bodies remained untouched for tens of thousands of years. Teeth sealed in sediment, bones broken, scattered, forgotten. No tools left beside them, only silence. When these remains were first unearthed, they seemed ordinary. Another jaw, another skull, another variation on a familiar form. They were catalogued, measured, compared, and placed neatly into existing times. Nothing about them suggested a fracture in humanity's story. Then something unexpected happened, began reading what was still hidden inside, not language, but molecular traces are damaged, fragmented, incomplete, yet unmistakably human. Yet material that had survived far longer than anyone once thought possible. What those sequences revealed was not gradual change, and not gentle branching. They revealed separation, deep, early, and persistent. Lineages that did not simply diverge and reunite, but remained isolated long enough to leave permanent marks in our DNA. This was not the split between humans and Neanderthals, not the well-known story of different species meeting at the edges of continents. This divide ran through ergations, before familiar cultures. Before the world was fully occupied for a long time, there was no way to see it. Fossils could not speak clearly enough. Stone tools told only part of the story, but DNA remembered what the landscape erased. And once that memory was recovered, it became impossible to ignore. To understand how profound this discovery truly is, we first need to step back to a time before genetics rewrote the past, and to what we once believed about the earliest chapters of human history before ancient DNA entered the picture. Early human history was reconstructed from what survived in the ground. Bones, teeth, stone tools, hearts, and fragments of shelter formed the foundation of everything researchers believed. From these traces, archaeologists and anthropologists built times, migration routes, and evolutionary relations. The method was careful, comparative, and slow, and for a long time, it seemed sufficient human fossils were arranged primarily by anatomy. Skull size, brow ridges, jaw shape, and limb proportions were used to define populations, and sometimes, species differences were interpreted along a spectrum. Some fossils appeared more robust, others more gracile. Some looked transitional, but most were assumed to belong to closely related groups, separated by geography or time, rather than deep biological distance. Similar technologies appeared across vast regions, often spanning thousands of years. Hand axes, blades, and later microliths follow recognizable patterns. When the same tool types appeared in distant places, it suggested shared knowledge or recent common ancestry. Cultiminavies of Finavan, early humans emerged in Africa and gradually spread outward in waves. As they moved, they adapted to new environments, but remained part of a broadly connected population. Even when groups became isolated for a time, it was assumed those separations were short-lived in evolutionary terms. The prevailing view was that early human populations frequently mixed, preventing deep genetic splits from forming. When unusual fossils were discovered, they were often explained as local variants. A skull with archaic features might represent a population lagging behind others in anatomical change. A particularly robust skeleton could reflect adaptation to cold or heavy physical labor. These differences were rarely seen as evidence of long-term isolation. Even the existence of other human species was once minimized. Neanderthals were initially viewed as a rough, failed version of modern humans. Denisovans were entirely unknown. The idea that multiple human groups could coexist, remain distinct, and occasionally interbreed took decades to gain acceptance. And even then, these groups were treated as exceptions rather than part of a broader pattern. Geography also shaped expectations. Africa was seen as the primary engine of human evolution, with Eurasia as a stage for later dispersals. Within Africa itself, populations were often treated as part of a single, interconnected system. Environmental barriers, such as deserts, forests, and shifting climates, were acknowledged. But their impact on long-term isolation was difficult to measure. Using archaeology alone, time, resolution, by tens of thousands of years, might be grouped together simply because there were no better reference points. Without a precise way to measure relatedness, continuity became the default assumption. As a result, early human history was often drawn as a branching tree with shallow splits, lineages disbarred for long. When branches rejoined, it was assumed they did so easily and repeatedly. Humanity's early story was framed as one of persistent connection. This framework shaped textbooks, documentaries, and public understanding for generations. It was internally consistent and grounded in tangible evidence, but it carried an unspoken assumption that what looked similar must also be closely related, and that absence of evidence for separation meant separation, not by reinterpreting tools or fossils, but by measuring ancestry itself. And when it did, it revealed that the archaeological record had been hiding a deeper story, one that could not be seen in stone or bone alone. That turning point began not with a dramatic fossil discovery, but with a technical breakthrough in molecular science, and it would quietly dismantle some of the most basic expectations about how early human populations were connected. The shift began in laboratories, not excavation sites. For decades, geneticists believed DNA could not survive long after death. Heat, moisture, bacteria, and time were thought to destroy it completely. The deeper past, especially the Ice Age, seemed forever beyond molecular reach. That assumption proved wrong. In cold, dry, and stable environments, fragments of DNA sometimes persisted inside dense bones and teeth, not intact germs, but broken strands, damaged at the ends, chemically altered, and mixed with microbial contamination. At first, these fragments were so short and rare that many researchers doubted their authenticity. Early attempts were plagued by modern contamination and false signals. Progress came through restraint rather than speed. New cleanroom protocols were developed. Samples were handled in sealed environments. Negative controls became standard. Researchers learned to recognize the molecular signatures of ancient DNA damage, patterns that modern contamination could not replicate. Slowly, confidence grew that genuine genetic material from deep time could be isolated and studied. Teeth and the densest parts of the skull became key targets. Within these structures, DNA was sometimes protected from the worst environmental damage. Even then, recovery was partial. Sequencing ancient DNA was not about reading a complete genome, but about assembling a mosaic from thousands of small fragments. Each fragment carried limited information. Together, they could reveal ancestry as xenogenealogies analysis. Instead of focusing on a handful of genetic markers, researchers began comparing hundreds of thousands, then millions of positions across the genome. Patterns emerged not from individual mutations, but from how those mutations clustered across populations. One of the earliest surprises came when ancient humans were compared to living humans. Some fossil individuals, long assumed to be ancestral to modern populations in their regions, turned out to be only distantly related. Others carried genetic signatures that no longer existed in any living group. These were not ancestors in a direct line, but branches that had ended. As more samples were added, researchers began to see structures where none had been expected. Early human populations that lived within the same broad regions were not always closely related. Some had diverged far earlier than archaeology alone had suggested. Their separation was not recent nor easily explained by local adaptation. In Africa, gnomes from different regions showed deep splits that dated back hundreds of thousands of years. In Eurasia, ancient individuals sometimes belonged to lineages that had separated long before. Their remains were deposit. They reflected long-term isolation. Dating these splits relied on mutation rates and comparative modeling. By counting genetic differences and calibrating them against known times, researchers could estimate when populations last shared common ancestry. While the exact dates varied, the overall picture was consistent. Some early human groups had been evolving separately for periods comparable to the time separating modern humans and Neanderthals. What made this especially striking was that many of these populations looked anatomically similar. Their skulls did not announce deep separation. Their tools did not reveal hidden boundaries. Without DNA, there was little reason to suspect such profound diversion. The breakthrough was not a single discovery, but accumulation. Each new ancient genome added resolution. Over time, it became clear that early humanity was not a single loosely connected population. Spreading outward, it was a network of groups, some exchanging genes, others remaining isolated for astonishing lengths of time. This realization reframed familiar events into empty space. They were encounters between populations with long separate histories. Interbreeding was not just a footnote, but a mechanism that reconnected deeply divided lineages, sometimes briefly, sometimes with lasting impact. Ancient DNA did more than confirm or deny existing theories. It exposed blind spots. It showed that continuity in the archaeological record did not guarantee genetic continuity. It revealed that early human history contained divisions that had gone unnoticed, because there was no way to measure them directly. By the time dozens, then hundreds of ancient Gurans had been analyzed, the evidence pointed in a single direction. The first major genetic divide in early humans formed far earlier than expected, persisted across vast stretches of time, and shaped the populations that would later give rise to modern humanity. Understanding what that divide actually was and why it mattered required looking closely at what the DNA itself revealed. When researchers began to compare ancient genome across regions and time periods, a pattern emerged that could not be explained by gradual change or simple migration. Early human populations did not separate briefly and reconnect. Some of them split early and remained apart long enough to follow distinct evolutionary paths. The first major genetic divide did not align neatly with familiar species labels. It was not the boundary between modern humans and Neanderthals, nor between Africans and non-Africans in the later phases of human expansion. Instead, it appeared deeper in time within early Homo sapiens. Genetic data indicated that by roughly 300,000 to 400,000 years ago, human populations in different parts of Africa were already diverging significantly. These groups shared a common origin, but environmental barriers, shifting climates, and changing landscape reduced contact between them. Some populations lived in relatively stable ecological refuges. Others occupied regions repeatedly disrupted by droughts or glacial cycles. These conditions shaped not only culture and behavior, but also genetic structure. Was not by separation dactyrate through intermittent contact. While others became effectively isolated, this divide was not a clean split into two branches. It was uneven, with one cluster of populations maintaining closer genetic ties, while another drifted further away over time. This produced lineages that were genetically distinct, despite appearing anatomically similar. The DNA made it clear that early humans were not a single cohesive population, but a patchwork of groups with different histories. Evidence for this came from patterns of shared mutations. Some ancient humans shared variants that were absent in others, even when fossils came from similar time periods. These shared variants acted like signatures of long-term ancestry, pointing back to periods of separation rather than recent divergence. Certain anatomical traits appeared and disappeared unpredictably in the fossil record. Stone tool traditions overlapped in ways that seemed inconsistent with population replacement. The DNA showed that these patterns could result from contact between groups that had been separate for long periods, reemerging again. Interbreeding played a crucial role. When previously isolated populations met, their genetic differences did not vanish. Instead, they were layered. Small amounts of ancestry from one group entered another, leaving detectable traces that persisted for tens of thousands of years. These traces allowed researchers to reconstruct. In some cases, entire lineages disappeared, leaving no direct descendant. Their genetic legacy survived only as fragments within later populations. Ancient DNA revealed that extinction was common and survival was uneven. Many early human groups contributed to our ancestry indirectly or not at all. This genetic divide also reshaped times. Some populations once assumed to be ancestral, others previously overlooked, emerged as central contributors to later human diversity. The DNA forced a reevaluation of which populations mattered most in shaping modern humanity. Crucially, the divide was not absolute. It did not produce separate species in the traditional sense, in structure that could persist without visible boundaries. This challenged the idea that species labels alone could capture human evolutionary relationships. By revealing how early and how deep this separation ran, ancient DNA showed that the foundations of modern human diversity were laid far earlier than expected. The populations would have a long history of division, contact, and recombination. This realization shifted the focus of human evolution away from a single origin story and toward a dynamic process. Humanity did not emerge from one population marching forward together. It emerged from many populations divided and reunited across deep time, carrying with them the genetic echoes of their separation. It changes how we understand what it means to share a common human past. The discovery of a deep genetic divide in early humans does not fragment our species. It does the opposite. It clarifies how interconnected human history truly is, and how much of that history unfolded long before written records, agriculture, or permanent settlements. Ancient DNA shows that modern humans are not the product of a single continuous lineage. We are the result of repeated connections between populations that had once been separated for immense spans of time. Our shared ancestry is not simple or linear. It is layered. This has important consequences for how we understand human diversity. Genetic differences among people today are shallow compared to those that once existed between early human populations. Groups that lived hundreds of thousands of years ago could be far more distinct from one another than any populations living now. And yet, those ancient groups were still part of the same evolving human story. The DNA record also reshapes ideas about adaptation and survival. Traits that help humans thrive did not arise in isolation. They were often assembled through contact. When populations met after long separation, beneficial genetic variants could spread rapidly. Combining as modern human resilience is in part the product of these ancient exchanges. At the same time, ancient DNA reveals how much was lost. Many early human lineages left no clear descendants. Their cultures, languages, and identities vanished, sometimes without trace in the archaeological evidence that they ever existed. This realization adds depth to the human past, reminding us that survival was never guaranteed. These findings also caution against oversimplification. No single population can be identified as the sole source of modern humanity. The genetic divide shows that our origins were distributed, not isolation. Importantly, this perspective remains grounded in evidence. It does not suggest radical discontinuity or separate origins of modern human. Instead, it emphasizes structure within unity. Early humans were divided, but not forever. The boundaries between populations shifted, opened, and closed in response to climate, geography, and chance. By recognizing this complexity, ancient DNA allows a more accurate, more humane understanding of our past. It replaces static categories with dynamic relationship. It shows that being human has always meant belonging to a network of connections that extended far beyond any single group. The consequences of this insight reach beyond science. They influence how we think about difference, ancestry, and shared heritage, not by offering philosophical conclusions, but by grounding those ideas in the deep biological record of our species. To fully appreciate the significance of this shift, we must consider what it means for the way we tell the story of human origins itself. For most of its history, the human past could only be inferred indirectly. Bones hinted at form, tools suggested behavior, landscapes implied movement. But none of these could reveal how closely early humans were related, or how long they had been separated. Ancient DNA changed that quietly and permanently by recovering genetic material from deep time. Researchers gained access to a record that had never been read before. This record showed that the first major genetic divide in early humans, formed early, persisted across long spans of time, and shaped the populations that would later reconnect and expand. It revealed separation without visible borders, continuity without closeness, and unity built from long-divided party or anthropology. Ancient DNA does not tell us how early humans thought, spoke, or understood their world, but it tells us something equally fundamental: how their histories intersected, and how deeply those intersections mattered. The emerging picture of human origins is neither simple nor fragmented. It is structured, dynamic, and deeply shared. Early humans were divided by environment and chance, but never beyond reconnection. The genetic divide uncovered by ancient DNA is not a story of separation alone. It is a record of how separation and contact together shaped who we are. As ancient DNA research continues, the details will change, dates will be refined, but the central insight is unlikely to disappear. Human history was never a single path. It was a braided one, formed from many strands that split, crossed, and merged long before history began.